Hi, everybody. So let's continue on our discussion on how soil health indicators as impacted by crop species and management. Soil health really originated at Sanborn Field on the MU campus, where it's the third oldest research continuous site in the world. And to many, this is the grandmother of soil health. This is 80 years ago when MU scientists started putting together many of these principles that we talk about today. But let's get a little review on soil health. It is the three different soil health indicators: our biology, our chemical, and our physical soil properties. When they put these together, where we want to optimize our soil for the production of our crops and to give the best nutritional possibility to our animals and to the humans. Now, the biology, chemical, and physical properties are all interrelated. For example, when you increase soil biology, let's say through cover crops, you'll improve your soil physical properties, which in turn will improve your soil biological properties. And together, these will improve your chemical processes because many of our soil microorganisms will make different chemical nutrients more available. So you can see they're interrelated, but they also, if you destroy one of them or reduce, let's say through tillage, where you break up your physical properties, you will reduce your biology, and that in turn will reduce your chemical soil properties. So you can see how they're all interrelated. Now, we really think that the soil chemistry and soil biology, those soil properties point to good soil physical properties. And at the core of that is our soil aggregate or our aggregate stability. And these are soil particles that are held together by organic matter, also by the bacteria, the fungi, hyphae, and also the microbial glue like glomalin that's produced by the fungi that help hold these different particles together. And now you have pore spaces for water holding capacity as well as soil aeration, and this helps the plant grow better. And also more water infiltration. And we want to try to make sure we can improve this and have good soil aggregation. And here is a microscopic close-up of a soil aggregate, and you can see that there are many small aggregates that are put together for a larger aggregate. It's real important that these can stay together in case we have rain or any other kind of environmental factor. If they don't stay together and they just collapse, then we don't have our good water holding capacity or our soil aeration. Now, at Sanborn Field, we have 130 plus years of research on these different plots. And one thing that we wanted to look at is soil health. And this is based upon just fertility treatments. So let's take a look at soil microbiology, where we have no soil fertility, where we have soil fertility, but it's N, P, and K in lime, or we have 6 tons of manure per acre. And as you can see, it's stair-stepped with the soil microbiology biomass. So with the manure, and we may expect, because not only do we get nutrients, we also get a carbon source for our biology. When we have better soil fertility, we can also have better plant growth, more biomass. So this also gives us some more soil microbiology activity. Now, how does that translate to soil physical properties? Well, if you look over to your left, we have the same no fertility, and manure. And we see that with manure, we do have better soil aggregate stability. And we have seen this in our water drought response to many of our crops. So we are seeing this interaction between soil fertility and our soil health indicators. But what about crop species? Well, this shows the comparison between corn in the blue and continuous wheat in the orange, and the corn is also continuous corn. And on the x-axis, we have no fertility, full fertility, meaning NPK and lime, and our 6 tons of dairy manure. So let's take a look at corn first. Just like in the soil biology, we see a stair step. We see with no fertility compared to full fertility, a slight increase, but significant increase in water stable aggregate stability. But then we have manure, a much, much larger aggregate stability. Now, wheat, on the other hand, we don't see this big change from no fertility to manure. What really sticks out, though, other than that, is that it's always higher than corn. So this tells us in these very long experiments that there's a big difference between corn and wheat in the aggregate stability, which is very, very important for many reservoir properties. So we want to compare then corn and wheat and soybeans. Does that make a difference, that crop species, in our soil health indicators? Well, we do have some data from Sanborn Field that we can look at. And this is where we have a corn, soybean, wheat, with a double crop, red clover, green manure rotation. And we have those individual phase of that rotation of wheat, soybeans, or corn. Now, there in the red is our soybean phase. And we can compare it to wheat and the corn. And this is OC is our organic carbon. As we see, it's significantly less than both wheat and corn. There are epoxy C, which is a carbon source for the soil microorganisms. There really isn't much difference between the three species. However, there is less total nitrogen with soybean phase, which is a little bit surprising because it being a legume. And there are potential mineralizable nitrogen, which also is a end source that's available for the plant, is also less with soybean. But our water stable aggregate stability is significantly less between soybean and wheat. Really no difference in corn. And our respiration. Now, our respiration is much less in soybeans than the other two. And our respiration is an indicator of how active those soil microorganisms are. So we see that it is less with soybeans than either wheat or corn. And it could be related back to the lower nitrogen in the soil and also the lower carbon. So what we want to then look at, let's look at more in the short term. In the 4 or 5 years rotation, or the 4 or 5 year period, can cover crops make a difference? And so we're going to look at cover crops both as a monoculture and a polyculture. Can that make a difference? And the reason we're doing that is that we have shown that legumes do better when they're mixed with a cereal crop. And but do these also have the negative effect, possibly, on soil health indicators? More than a cereal crop may have the benefit for us. So let's take a look and see what happens. Well, this is where we have different cover crops, either by themselves or in a mix. So we have corn and soybean. The blue is corn and the orange is soybean throughout. And we have no cover crop. We have hairy vetch or Austrian winter pea grown by themselves. So here we have a winter annual legume cover crop that we follow it then with either corn or soybean. And what sticks out, first of all, is that soybeans is less in aggregate stability than corn in all three of these. And it really doesn't improve in soybeans much with hairy vetch or Austrian winter pea, whereas we get a little bit of increase with hairy vetch in our corn. But what we do find is that when we follow soybeans behind cereal rye, we see that aggregate stability during that summer is equal to that of corn. So now this cereal crop that we showed at Sanborn Field how wheat made such a big impact on aggregate stability, that just using that as a cover crop could make a huge difference. Well, what's really also interesting is that when we started mixing our cereal rye with the annual legume, we lost some of that benefit. Then when we mixed them all together, we regained some of it, but we did lose some of that benefit when we mixed it with the legume. Now, soil microorganisms, this is the total weight of what's out there. And what we found was that in soybeans, it was always greater than corn. And it did improve slightly with mixing it with other cover crops, but not a whole lot. But this is kind of opposite of what we had saw at Sanborn Field, where we saw the respiration or the activity of our microorganisms. So maybe what this is showing us then is that yes, there are a lot of microorganisms, but they're not very active. Maybe there's not enough carbon in the soil or nitrogen in the soil to sustain them, so they don't have that good activity, even though they are there. So our conclusion then is that our cereal cover crops can help overcome some of the negative impact from when soybeans are in the phase of a rotation on our soil health indicators. But now we have another question. What about the length and the complexity of a rotation and the interaction with cover crops? How does that change our soil health indicators with soybeans? Well, we have 5 different rotation treatments. We have either a continuous corn and a continuous soybean. We have a corn-soybean rotation, or a 3-year where we have corn, soybean, wheat rotation, or a 4-year where we again have that corn, soybean, wheat rotation, but this year with a full year of red clover, or that same rotation of corn, soybeans, and wheat with 2 years of alfalfa. If we put some perennial crops in here for a year or two, how does that make a difference as well as wheat? One of the questions I had was just having cover crops in a rotation, is that the same as having wheat in my rotation? So let's see. I want to state that we always planted green, whether it's into the cover crops or into our perennial crops like this alfalfa or red clover. We would terminate them after we planted them. We did see visual differences in our soybeans with our rotation lengths in our cover crops. The photo on your left shows soybeans in a 3-year rotation, and you can see the cover crop soybeans are slightly larger than the non-cover crop soybeans. However, those on your left, which are in a 5-year rotation, are quite a bit larger than the 3-year rotation soybeans. And we saw this consistently throughout the years. So what happens to aggregate stability? Well, we have blue is no cover crop. The orange is a cover crop, and we have either continuous corn and soybeans, a 2-year, 3-year, 4-year, or a 5-year rotation. And as you can see, first of all, the continuous soybeans, that the aggregate stability is less with or without a cover crop. We see a slight improvement with a cover crop, but not a lot. We go to a 2-year rotation, and we do see some benefit from that. Again, with the cover crop, it's greater with soybean, but still does not equal to the corn. Now let's compare with a 3-year rotation where we added a wheat into the rotation. Now we see both corn and soybeans having greater aggregate stability, which kind of tells us what we saw at Sanborn Field in the long run. And we see that, but the cover crop is still a little bit higher, even in our 3-year rotation and including the wheat. But we will, if we do compare the cover crop in the 2-year to the non-cover crop in the 3-year, we still see that having the wheat, that little bit longer period into the summer, makes a big difference on our soil health. But what's interesting is that when we have a perennial crop in there with its red clover or alfalfa, we see that the soil aggregate stability improves both with corn and with soybeans. So extending their rotation, making it a bit more complex, we can see that we can get greater aggregate stability in the soybean rotation phase. Mycorrhizae are one of those indicators for our fungi. As you know, mycorrhizae extend the rooting system of a plant, and it's often destroyed by tillage. And what we found was in continuous, a 2-year, and even 3-year rotations, we didn't see a big difference, slightly in the 3-year, but again, in the 4 and the 5-year, that's when we really started seeing some differences in mycorrhizae in both corn and in soybean. Again, the epoxy We at one time thought that was a very active carbon. It's not as active as we once thought it was, but it still is beneficial for the soil biology. And again, at starting about the third year with soybeans, we start seeing this big improvement that lasts throughout the 4 and the 5 year rotations. So POC, or that carbon that's in the soil that the soil microorganisms can utilize, is increasing when we extend that crop rotation. So our conclusions on this part is extending the rotation and utilizing wheat or perennial crops can mask some of those negative impacts on soil health indicators during the soybean rotation phase. And we improve the aggregate stability or that soil structure, we increase the biology, and we also increase the soil carbon. So our overall conclusions then is that crop species do differ in their soil health parameters, with soybeans being lower than corn or wheat. Cereal cover crops can help overcome part of this deficit, but what we've really found is that having wheat in the rotation is what really can make a big difference. Now, mixing the cereal crops with the annual legume can mask some of these effects and equal them to corn. Annual cover crops, so in combination with extended the rotation with wheat or perennials, can improve soil health indicators in soybean phase of a rotation. So we want to thank our sponsor, the United Soybean Board, for this. They have sponsored this research that we do now, and we are still continuing. I invite you later on, we'll be having other videos talking about some of the other factors of soil fertility and drainage, and maybe what's really going on in the field about why soybeans may also always have a little bit lower soil health indicators than what we would like. We'll talk to you next time.